Today my topic is a general overview of the genetic alterations in ALL. And at the end of this speech, I will extend our discussion to its clinical implication. Okay, ALL is a disease with a lot of difference between children and adults. And the discrepancy is best illustrated by this figure, in which it shows that cytogen abnormalities are very different across different age groups. For example, in infant ALL, ML translocation is very common. And favorable prognosis factors such as hyperdiploidy and translocation between ETV6 and RUNX1 are really common in young children. In contrast, unfavorable prognosis factors such as Philadelphia chromosome is really common in older adults, up to 30 to 40%. And newly diagnosed and newly found chromosomal changes such as intrachromosomal amplification of 21 with multiple copies of RUNX1, and also overexpression of CF2 related to translocation with immunoglobulin heavy changing or deletion, resulting in fusion between R2, P2RY8 and CF2 are also have occurred in different age groups, but incidence are relatively low. And the discrepancy in cytogen abnormalities across age groups are supposed to be one of the reasons for the differential prognosis between different age groups. And most of the ALL arise from B precursor lymphoblast. In contrast, TAL comprises small proportions of AL across all the age groups. And using a high-resolution SNP array, scientists have found deletion of several important genes. There's some distortion of slides, okay. And as shown here, Icarus deletion here occurs in up to 13% of all patients, but incidence can be as high as 64% in those with Philadelphia chromosome. And PAX5 deletion occur in 19% of all patients, and incidence can be up to 45% in those with Philadelphia chromosome. And this shows that the cyclin-dependent kinase inhibitor deletion, it occurs in up to 27% of all patients, and the incidence in Philadelphia post AL is 48%. In contrast, ETV6 deletion, which occur in 22% of all patients, is frequently seen in patients with ETV6 and RUNX1 translocation. So in this case, actually, ETV6 seems to be homozygous loss. And it is really uncommon in Philadelphia chromosome, Philadelphia chromosome positive ALL. And deletion with other genes such as RB and also other EBF1 transcription factors also happen in B precursor ALL, but the incidence are relatively low. Let's go deeper into the nucleotide level. So alteration in nucleotide level causing gene mutations has been discovered in several genes, in many genes in B precursor ALL, thanks to the next generation sequencing technology. But the incidence here shows that mutation in Icarus 1 and 3 is really uncommon, occurring only 3%. Compared to its deletion in this group of patients, it can be up to 13%, so that's very different. And PAX5 mutation occur in 11%. And the cell cycle related genes such as p53 and the cyclin-dependent kinase inhibitor occur in 6% of these age groups. And NRAS and KRAS each occur in 15% in this group of patients. And the free-3 internal tandem duplication and PTP11, as we have seen in acute myeloid leukemia, also occurs in B precursor ALL, and incidence about 5%. And JAK and the IL-7 receptor alpha mutation, which we will discuss in more detail later, also happens in this group of patients. There are also a lot of genetic alterations in T precursor ALL. As shown here, about 45% of patients harbor translocation involving T receptor genes. And the fusion partner include TXL1, TX3, TAL, etc. And these different fusion partners may be responsible for the differential clinical presentation or prognosis in this group of patients. MAL translocation in 7%, and SIL-TAL1 fusion due to interstitial deletion in chromosome 1 also seen in 25%. Other mutations such as 1011 translocation, episomal fusion between NUP214 and ABL1, and also cyclin-dependent kinase deletion is also very common in T precursor ALL as well as in B precursor ALL. And there are several activating mutations such as NOTCH1 and FRI3-ITD, interleukin-7 receptor alpha, JAK mutations. And also there are several inactive mutations such as WT1, LPW7, and loss of P10. Duplication of CMYB is also seen in 8% of patients with T precursor ALL. So overall, the genes involved in ALL are often transcription factors or regulators, cytokine pathway or signal transduction, which are important for normal B cell development and development of normal immunity, as well as the cell cycle regulators such as p53 and cyclin-dependent kinase inhibitor. And most of genes involved in leukemogenesis in ALL frequently encodes critical transcription factors and coding modifiers in normal B cell and hematopoiesis. For example, in this review, before the establishment of hematopoietic stem cell, there are several important transcription factors which are very important in lymphocyte development, and they are also commonly seen the mutation of these genes are also very commonly seen in ALL. For example, TL1, RUNX1, MLL, and HOX. After establishing a hematopoietic stem cell, there are also many factors involved. For example, ETV6, which is important for maintenance of survival of hematopoietic stem cell. ECARS, as shown here, is critical for development from hematopoietic stem cell to common lymphoid progenitor. And there are important lineage commitment factors such as NOTCH1 and MYB, important for T cell leukemia genesis, and also PAX5 here, which is important for final commitment of B lymphocyte. As we have shown, it is frequently deleted in BALL. NOTCH1 activity mutation deserves more attention because it occurs in a very high proportion of TALL. Moreover, this mutation causes constitutive activation of Notch pathway after cleavage and release of intracellular Notch by gamma secretase. So gamma secretase inhibitor has been implicated in target therapy in this group of patients. And the constitutive activation of Notch causes activation of MEK and HES1 and finally lead to P10 downregulation. And then there was activation of PI3K and AKT pathway. And another important loss of function mutation, LPW7, because it is important for clearance of intracellular Notch. And as to TAL, there's one pathway that's important to mention. That is PTPN2 and the JAK1 and NUP214 ABL1. And PTPN2 is a phosphatase which can abate the activation of JAK1 signaling and the NUP214 ABL1 fusion. So the deletion of this PTPN2 as seen in 6% of TL will theoretically cause activation of these pathways and through a step 5 phosphorylation. And the result will be activation of cytokine signal transduction pathway in a cytokine-independent manner. There is also a major pathway involved in B and T cell development and also in leukemogenesis. That is IL-7 receptor alpha and the CSLF2 plus JAK pathway, as shown here. CSLF2 overexpression or mutation has been implicated in so-called BCR-like ALL, which we will discuss later. For example, illustrated in this figure, CSLF2 mutation at 232 from serine to cysteine lead to homodimerization of this mutant protein. And it will be cytokine, it will be ligand TSLP independent. Again, JAK mutation plus overexpression of CRF2, which may be due to translocation, also cause cytokine-independent growth. As shown in this study, overexpression of CRF2 plus JAK2 mutation at R6A3 will render the BAF3 cells to grow independently of cytokine. Again, IL-7 receptor alpha, also as shown here previously as a heterodimer of CRF2. The mutation may involve transmembrane region as a point mutation or insertion at the transmembrane region. And the mutation will cause also cytokine-independent signal transduction. In this S1A5 to cysteine mutation, it will cytokine-independent but still require dimerization with CRF2. However, in the transmembrane insertion, it will become cytokine-independent as well as CRF2 independent. They will self-dimerize. So here, again, these are very important pathways. CRF2 mutation, IL-7 receptor mutation, both are gain-of-function mutation, and both involve the addition of cysteine. The result is step-5 phosphorylation and constitutive cell growth. And they are also something different. For example, CRF2 overexpression is related, has been shown to be related to poor prognosis. And CRF2 mutation mainly seen in B precursor ALL, while IL-7 receptor alpha can be seen in both categories. Recently, by gene expression analysis, scientists have found a group of patients with a very similar global expression with BCR-ABL1. However, their cells did not harbor BCR-ABL1 Philadelphia chromosome, and they call this group as BCR-ABL1-like ALL. Subsequent study has proved that both BCR-ABL and the BCR-ABL1-like ALL had very poor prognosis compared with other ALL. And further analysis showed that both groups actually harbor at least 80% chance to have a deletion in at least one critical gene involved in B cell development, compared with only 36% deletion in other B cell ALL. So the similarly high incidence of this kind of genetic change between BCR-ABL1-like and BCR-ABL1 positive may partially explain the similar global expression profile between these two disease categories. An epidemiological study have shown that in childhood ALL, BCR-ABL1-like disease consists up to 15% of patients, compared with only 3% with BCR-ABL. And subsequent epidemiological studies show that actually BCR-ABL1-like disease can be up to 21% in adolescents and even up to 25% in young adults. So it seems that in young adults, there has been about half of the patients that harbor either BCR-ABL or BCR-ABL-like. And recently, there are high-throughput sequencing to analyze the genetic changes in this group of BCR-ABL1-like disease. And the scientists have found that in this group of disease, there are about 50% of patients with CRF2 overexpression related to either translocation with immunoglobulin heavy changing or fusion with P2RY8 or rarely mutation itself. And about 30% of these patients actually have JAK mutations or fusion with BCR or PAX5. And also 12% of them have IL-7 receptor gene mutation. 10% of them have free-3 internal 1010 duplication. And 8% has EBF1 and PTFR fusion or NUP214 and ABL1 fusion. And in rare cases, there are also several different genes fused with ABL1. So the implication of this finding is that JAK inhibitors, as we shown previously, is important to inhibit the signal transduction in CRF2 patients, JAK mutation, IL-7 mutation patients. And free-3 ITD inhibitor may be important for free-3 ITD. And also imatinib is important to abate the signal transduction pathway involved in translocation of PDGFR or AB1. So finally, my conclusion is gene alteration ALL frequently involves translocation with TCR or IGH, consistent with epi recombination during lymphocyte development, and it frequently involves lymphocyte development genes or pathway consistent with a blockade of their differentiation or either augmentation of their proliferation. And there are also frequent aberration in cell cycle regulator. And the implication is target therapy, because JAK inhibitor may be useful for CIF2 IL-7 receptor and JAK pathway. Gamma secrete inhibitor may be useful for NOTCH1 mutation. And imatinib may be useful for a translocation involving either ABL1 or PDGFR. So this is my talk. Thank you.